Ja, meine sehr geehrten Damen und Herren, liebe Kolleginnen und Kollegen, liebe Gäste hier vor Ort und die uns auch per Livestream zusehen. Lieber Herr Professor Beul und liebe Familienangehörige und Freunde, ich darf Sie sehr, sehr herzlich zu unserer heutigen Antrittsvorlesung von Professor Beul an der BOKU begrüßen. Und besonders begrüße ich Herrn Altvizerektor Josef Glössl. Schön, dass du da bist, Josef. Ich begrüße auch sehr herzlich den Leiter des Instituts für Bioverfahrenstechnik, Herrn Professor Gerald Striedner, und die Stellvertreterin, Frau Privatdozentin Astrid Dürauer. Herr Professor Striedner wird uns heute durch den Nachmittag führen in Vertretung von Professor Matanovic, der leider erkrankt ist. Ja, ich begrüße auch sehr herzlich Herrn Dr. Gengenbach. Er ist ehemaliger Doktorand von Professor Beul. Herr Dr. Gengenbach, herzlich willkommen bei uns. Und als Wegbegleiter von Professor Beul begrüße ich Herrn Dr. Niegut und Herrn Dr. Wessling. Herzlich willkommen, beide Herren. Ladies and gentlemen, dear colleagues, a very warm welcome to you here at BOKU and all over the world. We have come together today to celebrate the inauguration lecture of our dear colleague, Professor Johannes Beul, our new professor for downstream processing. The lecture will then be in English, but please allow me to switch back to German now. Lieber Herr Professor Beul, wir sind also heute hierher zusammengekommen, um Sie sozusagen offiziell bei uns an der BOKU sehr herzlich willkommen zu heißen. Sie haben Ihre Professur ja im September des letzten Jahres angetreten und ich denke, in der Zwischenzeit sind Sie so richtig heimisch schon geworden an der BOKU. Ich traue mich, das zu sagen und Sie nicken, das freut mich. Ihre Professur für Downstream Processing ist ja eine zentrale Professur für die Biotechnologie und damit auch für die BOKU. Denn die Biotechnologie ist nicht nur ein sehr, sehr starkes Kompetenzfeld unserer Universität, sie ist auch eine wichtige Technologie der Zukunft. Das hat uns ja gerade die Corona-Pandemie in den letzten 3 Jahren gezeigt. Die Biotechnologie hat in dieser Zeit eine immense Bedeutung erlangt und bei der Eindämmung der Pandemie ganz wesentlich geholfen, nämlich durch die Entwicklung von Impfstoffen insbesondere. Und das ist ja heute in aller Munde auf Basis der mRNA-Technologie. Diese Impfstoffe haben sich bekanntlich als sehr wirksam erwiesen und werden in vielen Ländern weltweit eingesetzt. Und die Biotechnologie hat auch bei der Diagnostik eine wichtige Rolle gespielt. Es wurden PCR-Tests und Antigen-Schnelltests entwickelt, um eben Infektionen mit dem SARS-CoV-2-Virus nachzuweisen. Und schließlich arbeiten die Biotechnologen auch an der Entwicklung von Therapeutika, um eben schwere Verläufe von Covid-19 zu verhindern oder zu behandeln. Und bei all dem, und da gehe ich jetzt nicht im Detail darauf ein, aber viele, die daran beteiligt waren, sehe ich hier im Publikum sitzen, war und ist auch die BOKU an vorderster Front dabei. Und das freut mich ganz besonders. Insgesamt hat also die Biotechnologie in den letzten Jahren gezeigt, dass sie ein entscheidender Faktor bei der Entwicklung von Technologien und Produkten zur Eindämmung einer Pandemie ist. Und die Forschung und Entwicklung der Biotechnologie wird ganz sicher auch in Zukunft eine sehr wichtige Rolle spielen, damit eben auf allfällige weitere, wir hoffen, es gibt jetzt eine Pause, aber auf allfällige weitere Pandemien und andere Herausforderungen im Gesundheitsbereich reagiert werden kann. Meine Damen und Herren, die moderne Biotechnologie hat aber nicht nur in der Medizin Bedeutung, sondern auch in vielen anderen Gebieten, insbesondere in der Landwirtschaft, im Umweltbereich und in der industriellen Produktion. Und es ist deshalb auch kein Wunder, möchte ich sagen, dass auch unsere Studierenden das erkannt haben. Im Fach Lebensmittel- und Biotechnologie haben wir auch einen besonders großen Zulauf. Im Herbst hatten wir sogar ein Plus von sage und schreibe 50% bei den Erstsemestrigen. Ja, das freut mich ganz besonders. BOKU-weit war es ein Plus von 8%. Und die Jobaussichten unserer Studierenden sind ja auch wirklich glänzend und ich komme gerade auch erst von einer Besprechung mit einem potenziellen Arbeitgeber, Arbeitgeberin und mit verschiedenen Stakeholdern. Und da kann man wirklich sehen, dass unsere Absolventinnen und Absolventen sehr, sehr gefragt sind und dass man eigentlich sprichwörtlich sagen kann, sie gehen weg wie die warmen Semmeln. Lieber Herr Professor Beul, Sie sehen, Sie werden gebraucht bei uns an der BOKU. Und wir freuen uns deshalb auch sehr, dass wir mit Ihnen einen exzellenten Forscher und Universitätslehrer im Bereich des Downstream Processing gewinnen konnten. Wenn man in Ihren Lebenslauf schaut, ist man beeindruckt. Professor Striedner wird uns sicherlich noch etwas mehr über Ihre Vita erzählen. Ich möchte jetzt nur eines erwähnen, nämlich, dass Sie vor kurzem mit dem renommierten DEKEMA-Preis ausgezeichnet worden sind. Und zwar für Ihre herausragenden Beiträge zur Produktion und Isolierung von Wirkstoffen mittels pflanzlicher Systeme. Also zum Beispiel, wenn es um monoklonale Antikörper für Krebstherapie oder um Impfstoffe geht. Die Optimierung der Produktions- und Reinigungsprozesse für diese Wirkstoffe, so heißt es, ist bis jetzt oft nach dem sogenannten Trial-and-Error-Prinzip erfolgt. Und diese Prozesse sind aber jetzt durch Ihre Forschungen viel vorhersehbarer, effizienter und kostengünstiger geworden. Lieber Herr Professor Beul, ich freue mich und die Zuhörer und Zuhörerinnen ganz sicher auch schon jetzt darauf, dass wir Näheres aus Ihrer Forschung hören, dass Sie uns ein paar spannende Einblicke in Ihre Forschungsergebnisse geben. Und ich darf Ihnen namens der BOKU, aber auch persönlich sehr, sehr herzlich zu Ihrer Professur gratulieren. Alles Gute und viel Freude und Erfolg bei uns an der BOKU. Und jetzt darf ich an Professor Striedner übergeben.
So, grundsätzlich war es nicht so geplant, dass ich diese Moderation mache. Von meiner Seite jetzt an den Diethard Matanovic, unseren Departmentleiter, wünsche ich gute Besserung. Ich hoffe, du kannst zumindest über die Übertragung dabei sein. Für mich als Leiter des Instituts für Bioverfahrenstechnik ist es natürlich eine große Ehre, das machen zu dürfen, weil Johannes ja bei uns im Institut angesiedelt ist. Und ich muss jetzt sagen, von der Bedeutung und Wichtigkeit, die dieser Professur zugemessen wird, könnte ich es jetzt nicht wirklich besser darstellen, wie unsere Frau Rektor das jetzt gemacht hat. Ja. Also es ist wirklich eine zentrale Position. Und was ich jetzt gerne machen würde, ist noch einmal ein bisschen zurück, weil es ist im Grunde genommen so, eine Professur ins Haus zu bekommen, ist eine sehr spannende Angelegenheit. Und das Berufungsverfahren an sich ist schon ein großer Aufwand und nicht immer sehr erfolgreich. In dem Umfeld, wo du dich beworben hast, war die Bewerberlage sehr gut, wirklich gute Kandidaten und du hast dich durchgesetzt. Also kann man auch davon ausgehen, dass deine Ausstattung, deine Expertise wirklich anerkannt wurde und dass du das sehr, sehr gut geschafft hast, das überzeugend rüberzubringen. Es hat dann natürlich diese Verhandlungen gegeben, da kriegt man von der Department- und Institutsseite nicht immer wirklich sehr viel mit, was denn gerade passiert. Ja. Am Ende war es dann so, wir haben die frohe Botschaft gehabt, es ist erfolgreich verhandelt worden und der Johannes wird unser neuer Professor. Jetzt ist es so, es ist eine große Freude von der Instituts- und Departmentseite und es bleibt dann aber trotzdem immer noch so etwas wie ein Überraschungspaket, weil der Kollege hat sich gut geschlagen bei der Präsentation, man hat erste Diskussionen gehabt, aber halt sehr auf kurzem Wege und vielleicht nicht sehr detailreich. Und am Ende stellt sich immer die Frage, was haben wir denn uns jetzt da eingekauft mit diesem Überraschungspaket? Ja? Und das würde ich jetzt gleich einmal damit hernehmen. Ein Überraschungspaket ist sowas und mit diesen Überraschungseiern verbindet man normalerweise was Positives. Und wie wir ja schon gehört haben, ist der Johannes schon eine Zeit lang bei uns und wir können schon einen ganz guten Rückschluss machen, wie denn dieses Paket aussieht. Und aus der Department- und Institutssicht ist es so, der Johannes ist ein unglaublich offener Charakter. Ja, es ist gegenüber den Kollegen, gegenüber Partnern in der Industrie, gegenüber den Studenten, ja, ausgesprochen motiviert. Und es macht Spaß, mit dir zu arbeiten. Das können wir mittlerweile schon mal wirklich so festlegen und sagen. Von deiner wissenschaftlichen Seite ist es auch so, dass es für unser Institut und das Department wirklich eine tolle Erweiterung und Ergänzung ist. Du hast deine Fähigkeiten im Bereich der Modellierung von Bioprozessen, du bringst für uns wirklich neu die Bioverfahrenstechnik im Pflanzentechnologiebereich dazu. Das ist wirklich neu für unsere Uni und das wird die Departments auch weiter verbinden, ja, weil natürlich Pflanzenbiotechnologie recht breit aufgesetzt ist, aber die Verfahrenstechnik immer ein wenig gefehlt hat. Also ich denke, dass das eine sehr, sehr schöne Kombination ist für uns, da noch effizienter und noch besser arbeiten zu können. Ich würde jetzt nicht sonderlich viel zu deinem Lebenslauf da von mir geben wollen, weil dass da viel dahinter steckt, das ist eindeutig. Ja. Ich möchte nur eine kurze Geschichte da noch zum Besten geben, was ich weiß von dir. Ja. Und das ist, wir haben gesprochen über die Habilitation. Ja. Normalerweise wird die Habilitation an einer Uni durchgeführt und ist sehr schön irgendwie vorgegeben, was sind die Bedingungen, wie kommt man dazu und ist ein recht gerade gezeichneter Weg. Ja. Du hast mir erzählt, wie es dir ergangen ist, weil du hast es über Fraunhofer gemacht und da hat es keinerlei Regeln gegeben und ich glaube, das ist dann schon eine gute Portion an Willen, dass man überhaupt dorthin kommt, sich zu habilitieren, weil sie, soweit du mir das erzählt hast, die sehr, sehr lange irgendwie mit ständig erweiterten Anforderungen konfrontiert haben und an und für sich zu keiner wirklichen Entscheidung gekommen sein, was denn genug ist. Ja? Also hast du auch eine gewisse Ausdauer da schon einmal wirklich bewiesen. Ja? Da es ja nicht meine Aufgabe ist, da zu viel Zeit zu verbrauchen, würde ich dann auch relativ schnell jetzt in den Vortrag gehen, der natürlich in Englisch sein wird. Wir werden danach natürlich diskutieren und für alle Fragen, also das ist dieser grüne Würfel und man hat mir gesagt, dass das meine Aufgabe ist, dann den jeweils Fragenden irgendwie zuzuwerfen, damit ihr dann auch entsprechend mit Mikrofon versorgt seid und dann bitte ich einfach bei Fragen dann zum nächsten weiterzugeben. Ja? Also wirklich, ich freue mich auf deinen Vortrag. Wir werden ein bisschen was hören über das, was du gemacht hast, was deine Pläne sind und wie du quasi die BOKU in Zukunft da mit deinen Ideen befeuern wirst. Gut. Dankeschön.
Ja, vielen, vielen Dank, lieber Gerald und Frau Rektorin, für die super Einführung. An der Stelle würde ich jetzt auf Englisch wechseln. Ich möchte mich aber vorher noch wirklich bedanken, dass ich hier sein darf. So switching to English, it's really an honor for me to be here today, to be at BOKU. This is really for me a dream coming true, and also it's such a big honor to have this audience here with me today, both in the lecture hall as well as online. So thank you very much for giving me the opportunity to be here today and be part of this. I just recognized the pointer is missing, so if you would have, or if it's somewhere here, at least I cannot find it. But that's not a problem. I'll just.
I saw it here.
Or maybe I'm just blind. Could also be. That was a well-prepared joke.
It's an old habit from my lecture.
We talked about this. So, thank you. Now the anxiety is gone on my end, so that's perfect. So when I was looking at the title, after having submitted it to Frau Toth, or Ms. Toth, I recognized, okay, it's probably not the best title because some parts are missing. There should be definitely bio in here, and there should be research in here because this is what we do also at the university, right? So this is probably the more appropriate title, but what can you do? The next thing I ask myself is, what is this lecture going to be? Because I never had any inaugural lecture yet. So I was wondering, what can I do? What are people expecting? And who's the audience? So I was trying to put myself in your shoes. And obviously, there's people around here from life sciences. So I was expecting, okay, you probably want to hear something about life sciences. But then again, there's also other people from a diverse set of disciplines that may also want to hear something about life sciences, but maybe in, say, a different or at a different level of understandability. And also, it's maybe not just the life sciences, but also some interest about the person. So who is this new guy, as we just heard from Gerald? Like, what is he doing, or what's his vision? And then also, of course, about the science and what we have in mind. So I tried to balance that when I prepared the lecture, and I hope it's all right. I think satisfying all of these requirements is not easy, but I'm happy to get your feedback on that. Also, before we start, a short disclaimer. So time is short, and I probably will simplify things to make my point. And if you have questions about something that I didn't go into detail or into very much detail, please feel free to ask me afterwards. And then what follows is really my personal perspective and opinion, so please do not hold any Boku representatives responsible for what I say. This is not synchronized with them, and I've been working with plants for quite a while, and therefore my perspective might be unintentionally biased. I apologize for that. The first thing I would like to make very clear here is what you're going to see is not my work, it's the work of my past and present team. So I think in science you are nothing without your team, and therefore my heartily thanks go to my former and present team members. Accordingly, also the thanks go out to the partners in academia and industry that have cooperated with us for so long, and also the funding agencies and industry partners that have supported our research. Now, in the next 40 minutes or so, we will have a short walk through what I would call bits and pieces, because I thought, okay, now I'm a professor. In addition to the teaching, I can do some preaching, and this is what you're going to hear first, because that will then allow me to sweep away all the things you didn't like about the preaching with some science. And this is going to be the next part, so we will be talking about plant molecular farming and how we may be able to make biopharmaceutical manufacturing a bit more sustainable, or at least add the zoo of different production systems. Then we will talk about bioprocess modeling, which is really a topic that's close to my heart. And I'll finish with some product examples to show you that this is something that can actually be turned into application. And we'll briefly discuss integrated biomanufacturing, both on the material side, but also on the data side. Now let's start with the bits and pieces. So the first thing I think that's really important is we should be humble and thankful for what we have. So it's really the basic things. And if you're applying for some project, your proposal is being rejected, okay, that feels bad. But in fact, you're not being hunted by some saber-toothed tiger that's going to eat you. Most of us can feel very safe these days. So this is really something you can, even if things go wrong, you can say, okay, it's not that bad, right? So we are in a good situation all in all. And also we should be respectful for all the people that actually facilitate the work that we do, because working at a university, I think, is it's a privilege, because there's many people, hardworking people, that give their tax money to us to do something good for society, and we should keep that in mind whenever we do our work. However, being humble doesn't mean that we shouldn't strive for something big, like climbing the Mount Everest or so. We should still be negotiating hard for our vision, for our science, and we shouldn't be afraid of the big challenges, because we can break them down into small pieces and we can solve them as a team. And that, again, should sort of relieve us of some fear or fright. The next thing is very important, I think, at a university, and that is think for yourself, question authority. Me being an authority, that means you can question myself, and that goes out to the students, so you can question me as a professor. And that is something that takes energy, because it causes some sort of friction, right? If you're being questioned, then you'll have to think about answers. And it's maybe not something that you can do every day, all the time, but from time to time you should do that. And I think there's 3 groups of people that you should especially question. And one group is this very nice set of people. It's your bosses. So this is my bosses. And I should question them time and again. That doesn't mean I should be disrespectful, but I should question them. And at times it might be easier to ask for forgiveness rather than to ask for permission. The second group is your bubble, your peers, the people that are around you. You think, yeah, we did it, chromatography, this is how we do it, like this is how we've always done it. But then maybe there's a new person that doesn't know that everybody is doing that way and can solve a problem because they're doing it another way around. And then the last part is probably the group that is most difficult to question, and that is your friends and family, including yourself. So you should question what these people tell you, because most of the time they mean good, but maybe they don't know what you want. So this is also something that you should think about. And then coming maybe back to more science-related topics, I think it's important to support, reach out, and connect. And that does include reaching out into schools. I'm not saying that as professors we are school teachers, but we should connect with teachers at school to understand what is going on, what their problems are, because these problems are probably going to become our problems in a couple of years. And we should go into schools, talk with pupils there, and highlight what are the strengths, the opportunities, for example, when studying biotechnology, biology, chemistry, physics, etc., so that they are not afraid of what's coming along. What I've heard just recently from a colleague from the US who came to Germany, and I'm not saying Austria is like Germany, but who came to Germany and said before that she was very open and thought, okay, I'm going to pursue a career in science. But when entering school, she constantly heard back, oh, wow, you're going to do science? Like physics and chemistry? And then as a girl? Ooh. And that is creating a permanent atmosphere of fear. And that is something I think that we should resolve. And connecting with schools can take time and can be a laborious process, and I'm speaking from experience. So I established a collaboration back at Fraunhofer with my previous high school, and it took us 2 years to set that up, but it can be done, right? And I think this is what you need a long breath. Also, what we should think about is new topics, new content in our lectures, in our teaching. And what is in everybody's thought is probably topics like digitalization. Personally, I don't like that term, because actually what we need is data literacy. That is, we need to know how we can handle data, how we can handle information, and what we do with information. And that is a topic I think that we should very deeply integrate in into our teaching and courses. Also, I think we should be the vanguard or even the spearhead in that, and not be the rear guard. For example, when looking at companies like BASF that implement augmented reality in their on-site training and lab courses, I think this is something that we should implement here at BOKU as well, and that will take resources and a structured approach to do that, but I think it's going to be very important. Along that line, and then maybe from a student perspective, it's easy to tell, okay, you need to do this, and in addition you need this, and in addition you need this. So the other question is, where can we disinvest? Like, what are the topics that we don't need anymore, or at least not in that depth? I know that this is a very, say, tricky topic to discuss, because mostly teachers like what they teach, and so giving up something that you've taught for quite a while can be difficult. But if you look at the situation and at, say, the turnover time of knowledge, then it might become more necessary than ever to, in a more frequent way, look at what we have been teaching and replace old stuff. Like, for example, the knowledge about how to make arrowheads. It was probably indispensable 5,000 years ago, but hardly anybody knows about it today, just as an over-exaggerated example, of course. But we should think of the contents of our teachers and update them regularly. And then regarding support, I think if you look at it from a biological perspective, once you've reached the status of a professor, you're kind of old. And from a biological perspective, your brain has passed the peak of its performance, and therefore we should support the younger generation and giving them opportunities. And that means getting them from the status where they are listeners to the status where they are the people that people tell something. I'm very happy that you are here, Ben, today, because this has happened, I think, to you. So this is very nice to see. And in that context, I think it's important to step across the boundaries, the borders of your discipline and connect with people from math, from chemistry, from physics, from informatics. And again, that can be done. It might feel awkward at first because, speaking from personal experience, whenever I'm with the downstream community, I'm the plant guy. And whenever I'm with the plant community, I'm the downstream guy. But this is how it is. If you're crossing borders, then perspectives change. And the plant community brings me to the next topic, which is plant molecular forming. And I don't tell you anything new if I say that plants are one of the major sources of all human food, animal feed, and a large fraction of our pharmaceutical products, including building materials as well. Additionally, all of you know about climate change and how devastating it is to our natural resources and ability to produce or to sustainably produce new products. At the same time, what we see is an increasing demand for plant-derived protein for several applications. One is human nutrition, for example, to replace animal-derived proteins. In addition to that, many people are trying to replace animal-derived components in feed, for example, to replace fish meal in fish food. Furthermore, what we need is bio-nanomaterials or nanomaterials in general for new functionalized devices and equipment, and lastly, an urgent need for new pharmaceuticals. And this is mostly what I will focus on today, the production of biopharmaceuticals in plants. There's one thing I want to make very clear, that I'm not saying plants are the best production system there is. What I am saying as bioprocess engineers, we are, say, a company, and the products, the proteins, they are our customers, and the customer's always right. And so what we should do is we should pick the production system according to what the product needs. And there's obviously very good reasons to use microorganisms for certain products, mammalian or insect cell culture for others, but there can also be very good reasons to use plants, and I'm going to show you some examples of those. Stepping back out of this lecture for a moment and looking from, say, maybe a rectorate perspective on this setting, I think we have a very good opportunity and a very nice environment here now at the Department of Biotechnology, covering all this technology and providing services for partners in the industry and in academia. And showing these faces doesn't mean it's just them, it's a team behind each and every person making possible what we see here. Now, what can be the motivation to use plants as a production system? There are several, and maybe I just pick out some, and one is it's a very simple system. So if you want to produce or if you want to cultivate plants, that is fairly straightforward. You don't have to work in a sterile environment. You can use very simple fertilizers, etc. Another option or another perspective is it's safe because human pathogens are unable to replicate in plants. So it's a very safe system. And something that we will be hearing about later is we can reuse the residual biomass from plants for additional products and other processes. Obviously, plants do also have their drawbacks or their limitations. And some of them are that we have a high particle burden when we extract proteins. There's a lot of host cell proteins present, so impurities present in the preparation, including pigments. And on the next couple of slides, we'll see how we can resolve actually these problems. Before we do that, I would like to briefly introduce the different types of plant-based production systems that we can use, because there's quite a number of them. For one, we can use intact plants for the production of pharmaceutical proteins. That is, for example, genetically engineered, genetically modified plants. We can decompose the plants and use tissue culture, also using transgenic cells. We can further decompose it using plant cell culture in suspension, which is very similar to what we know from, say, E. coli-based or mammalian cell-based processes. And we can use cell-free systems, that is, decomposing even the cell. For plants, you can use any of these technologies with so many different plant species, but in addition to that, you can use transient expression as well. Transient expression is something that you may know from other systems, and it is the production, not in transgenically modified plants, but in cells that do not encode for the product themselves, but are being transfected. And for that, in plants, you can use agrobacteria, which is a soil-borne bacterium that has the natural ability to transfer DNA into plant cells and then trigger the formation of a product. We can use that technology in intact plants, but we have also developed a technology that allows us to do so in suspension cells, and we call that plant cell packs. What they are is, in fact, you take cells from a suspension, you remove the liquid medium and cast them into such 96 well plates. And on the next slides, we will see how we can use that to produce biopharmaceutical proteins and why that is important. Before we do that, just a very brief history about plant molecular farming, which is a very nice example of the Gartner hype cycle. That is, in the early 80s, the technology has been developed and substantial hopes were put in it because back then titers in mammalian cell culture were moderate, not as high as they are today, and there was a fear of a global shortage of biopharmaceutical production capacity. Reaching a peak in the hopes, you can see that major companies like Bayer joined into this technology, but shortly after, the environment changed. That is, you needed GMP-conform, GMP-compliant production of the pharmaceuticals, but little had been done about how to manufacture biopharmaceuticals in a G-compliant manner in plants, and therefore many of the companies involved in the topic went bankrupt. Bayer went out of the technology as well. And what happened afterwards was the maturation of the technology. So people sat down and really thought about how can we improve the manufacturing processes themselves and not just test different types of plant species, different products, etc. And since then, I think we have seen the technology substantially mature, and just as of last year, we've seen the first biopharmaceutical produced in plants getting regulatory approval. Now, how does such a process look like? Well, the major difference to conventional processes is really at the start, where we use the plants, which require that we seed them, that they are being cultivated, for example, in a vertical farming unit that is in several layers above each other. And if we're using transient expression, what we need in addition is obviously we need a fermentation for the agrobacteria, which can then be infiltrated into the plants, for example, by submerging the plants into the agrobacterial solution, applying a vacuum, releasing that, and the plant will soak up the bacteria, so to say. After that, after we have produced the protein in the plants, the process is somewhat similar to what you know from other bioprocesses. So we will have some extraction. The extractor will look a little bit different. We're going to see that in a couple of slides. And then you have a battery of clarification steps where we remove all the particles, the dispersed particles, and purify, for example, via chromatography. So the end of the process is very similar to what we know from other steps. And what I would like to show you in the next slides is how we contributed to this overall process. So at first, what we did was that we have created a modular vector system that allows us to test different genetic elements, for example, promoters, UTRs, different genes and gene variants, so that we can screen new biopharmaceuticals more effectively. Including in this modular system, we have the ability to direct a product to certain compartments within the cell. And the plant cell has a large number of these compartments, for example, the vacuole or the apoplast. The advantage of these compartments is that we can physically separate, for example, a toxic product from its potential molecular target and thereby protecting the expression host, preventing that the plant, for example, is damaged by producing a toxic protein. Because we can screen so many different genetic elements and test so many different compartments, screening in high throughput is very important. And using the plant cell pack technology that I showed you before, this is possible. Before that, it was kind of a hard work, and screening was really a limiting factor. So in this plant cell pack technology, what we can do is we can screen, for example, on a single plate, 96 different constructs. Before that, before that technology, what you had to do is you'd have to take such a leaf and manually inject the agrobacterial solution into that leaf time and again. So each of these little dots that you can see here is an attempt to get the bacterial solution into that plant. If you are kind of well-versed and you have mastered that this craft work, then the expression might look like this. So you have produced a red fluorescence protein in here. This is looking okay. However, if you're not so familiar, it might look like this. And as you can see, the expression is rather heterogeneous. And if you would test, for example, how much of a product is being formed, the results could be very variable. In addition to that, it's not enough to just express the protein, but you also have to puncture the leaves to get some sample to analyze the quantities of product that are being produced. So using this plant cell pack technology, this can be very much simplified. You have a 96 well plate format. You can just add the agrobacterial solution. You can have them drop onto these cell cakes. You centrifuge it. Everything is being removed. You incubate this plate. You can put it into a 96 well plate reader. You get your data out and can proceed with this, with the analysis and optimization. So a very straightforward process. The other benefit of the system is that it's fairly scalable. That is, the results that we can get from this 96 well plate translates very well into a setting, for example, where we have this vertical farming unit in intact plants. And what you can see here on the middle part is the expression levels of 3 different monoclonal antibodies, either in this plant cell pack, so on the right-hand side, or in different plant species. And even though the absolute expression levels might differ slightly, the relative expression, that is M12 is being expressed substantially more than 2G12, than 2F5. This is conserved across all the systems, so the results are fairly transferable between the plant cell packs and intact plants. Now, once we have produced the proteins in the plants, we have to get them out. And to do that, one approach is we can use these oversized kitchen blenders. The nice thing about them is that they can easily extract proteins. That is, you can see here, for example, a red fluorescence protein fairly constantly extracted, also an antibody fairly constantly extracted. However, the size of the equipment can be rather large. So at this point, we shouldn't be shy to learn from others. Here in this context, for example, learning from food processing, if instead of this large-scale blender, we use such a juicer, we can substantially reduce the size of the devices, and we can integrate both liquid extraction, what you can see here, you can get the liquid out, and at the same time you can get the solids out. So you have an integrated process that doesn't require to add buffer, which increases the volume, but actually reduces the volume of your equipment and therefore saves costs. And if you compare the two technologies, each of them have advantages and advantages and drawbacks. As I said, the blade-based extraction is very effective in that you can recover most product. The screw press might not be as effective, but it can save costs in terms of investment and the infrastructure. So you have to balance which of these technologies you want to use. Regardless of the technology you choose at the end, what you need to do with either this extract or this extract, which can be fairly viscous, is you need to clarify it. That means you have to remove all the particles dispersed in the extract. And this is a process step that is often overlooked, because in the lab you can just go to a centrifuge, put your sample into the centrifuge, get the sample out, and you're done with it. But at large scale, it's kind of difficult to centrifuge a cubic meter of extract. So if you want to develop a scalable process, you should look into filtration technology, but also maybe into additives. And on the next slide, I'm going to focus a little bit on these flocculants, which can do a very nice job in clarifying a suspension. Now, what is flocculation? Flocculation is a process in which we try to cross-link the dispersed particles in our plant extract, for example. Typically, they are negatively charged, and what we want to do is we want to add a polymer that is cross-linking these particles. This can often be achieved by adding positively charged polymers, such as polyethylene imine, but the efficacy of flocculation does depend on several parameters, like the pH, salt concentration, specific type of polymer to be used, particle concentration, etc. So what you need is a substantial screening effort to identify the flocculant that is doing a good job. And what you're seeing here is different plant extracts. Most of them look greenish, that is, the polymer isn't doing anything, but you can see some that are looking red because the green color from the plant juice has been precipitated and only the red color of the dispersed protein is still visible. And what we can do in this context, if we combine, for example, flocculation with so-called filter aids, filter additives, that are, for example, cellulose fibers, we can substantially improve the filtration capacity, that is the volume that we can pass through a unit area of filter several-fold. If we start without any flocculation and without any filter additives, the starting filter capacity here is somewhat of 20 liters per square meter, which is very bad. If at that point then we add flocculants that can help us form larger particles and combine that with cellulose-based fibers, choosing the right fiber concentration and size, what we can get to is filter capacities of more than 1,000 liters per square meter. And this substantial improvement can really reduce the costs for this, this downstream step, which has been a major cost driver for plant-based processes. However, as many others, this doesn't come, say, for free, and there can be a catch to it, and that is because we're using highly charged polymers. These polymers may bind to a chromatography resin, effectively blocking the resin and therefore preventing us from using that specific resin as a subsequent capture step in our purification process. So again, we have to balance whether using a flocculant is useful or not. This is just some pictures to give you an impression about how filtration processes can look like at bench scale, at lab scale, and at pilot scale. At best, you're using the same filtration devices and settings all the time to ensure that you have a good scalability of your process. Now, if we have clarified the plant extracts, we still need to purify the product, because if we look at these images, what they show is protein stained in blue, and this looks like, oh, there's a lot of protein in here. And yes, this is not our product, and this is not our product, but this is our product. So we need to get rid of most of the protein. What is highlighted here with the green arrows is Rubisco, the most abundant protein on Earth, and we have to get rid of it to be sure that we can supply a pure product. The way to do it is we can use polymer-induced precipitation, or we can use pH-based precipitation, for example, by reducing the pH down to 4 or at least less than 5. Most of the host cell proteins, that is the plant proteins, will precipitate. As you can see, this major protein has disappeared at a pH of 4, whereas our product is still present as a pH of 4. If the product is not stable at low pH, what we need to do is use some other techniques. We can use heat to achieve a similar thing. So if we heat the extract, what we can see, the plant-based proteins, they disappear from the solution. Only our product remains in solution. Again, this is a major, say, intervention into the process because the product may not be heat stable, and therefore a more gentle technique might be advisable. And that could be, for example, ultradiafiltration, that is a membrane-based separation process where we separate proteins based on size. And this is what we can see here. We have the plant extract, we filter it, we clarify it, and then we apply a membrane. And in the retentate, what we can see is that many of the plant proteins do not pass through the membrane, but are being retained on the membrane, whereas our product that is being colored red here is able to pass through the membrane. What you can see is that some proteins that are small or smaller than the proteins that we find in the permeate seem to be retained. And this is also the reason why ultradiafiltration is such a nice process for for the separation of plant proteins, because most of the plant proteins will form oligomers. That is, they aggregate, or they don't aggregate, they interact with each other and form bigger bigger protein complexes which are easier to separate. And if you account for this oligomerization, which you can find is that there is a nice window of opportunity down here that is of a size between, say, 10 to 50 kilodalton, which is a typical size for pharmaceutical protein, where there's essentially no plant protein present. Now, this is all done without chromatography. Then why do we need chromatography? Well, we need it to get to the final purity that is required for biopharmaceuticals. And in that context, I would like to come to the next topic, which is bioprocess modeling. To give you a very brief introduction about chromatography, in chromatography we have a packed bed column, often, that looks like this. And the column is characterized, for example, by the mode of operation. So we can either bind a protein to that column, or we can try to have everything else, so all the impurities bind to the column and our product to be in what is called the flow-through. On the next level, the column can be characterized by the packed bed, so the resin particles that form this bed, and by the porosity of these resins. At the molecular level, the chromatography is characterized by the proteins that we are interested in and their ability to bind to the ligands of this resin. The nice thing about chromatography, here you can see some of the chromatography columns at mid-scale, at very small scale, is that we can achieve very high purity of a product. What you can see here is the large and the small chain of an antibody that is, for example, being used in HIV treatment. And you can see that there's nothing else but the large and the small, or the heavy and the light chain of this antibody. So this has been achieved by chromatography. However, to get there is kind of difficult because we have so many different options in chromatography. That is, we can use so many different types of resins, we can use so many different types of conditions, for example, different pHs, different salt concentration, different ligand modalities, that identifying the most suitable path to get to our purified product is rather difficult, even if we're using automated lab equipment. So at that point, it would be very helpful to have a model that can predict the separation of proteins, including the impurities that are present, for example, because we have produced the protein in plants, and our product itself. And what we do in this context is we try to implement a hybrid modeling approach. And let me briefly walk you through this. So what we do is we take so-called training data, that is, proteins for which we have both experimental data about their binding behavior to the different chromatography resins, and we can calculate so-called descriptors for these proteins. I'll explain what a descriptor is in one of the next slides. From these experimental data, we can get what is called isotherm parameters. This is essentially the values that will dictate how the binding behavior of proteins looks like. And what we do then is we take the descriptors and we take the isotherm parameters and identify correlations among them. To verify that this model, this correlation that we have established is somewhat reliable, we take an additional set of proteins, the test set, and we check if the predictions that this model makes about the isotherm parameters of the protein does match with the experimental data that we have for this for this test set. Ultimately, once we have verified this model, we can apply the model to predict isotherm parameters for proteins for which we only have descriptors and nothing else. That is, we can make a priori predictions about how these proteins will ultimately separate in a chromatography setting. And maybe to use an analogy, if this approach seems a little bit complex, you can think of the proteins as different fruits. And you can think of the experimental data as creating, for example, a fruit cake from the fruits, and you can think of the descriptors as some external appearance, for example, the color of these bananas. And what we do then is, based on the color and based on the experiment, make a correlation and predict how well the taste of these cakes is going to be. We check with the test set that our rating of the taste is okay, and then just based on the color of another fruit for which we have never baked a cake, we make predictions about the taste and ultimately how well they are being sold in a bakery. Now, what do we need for this approach? What is required to get there? And it's not a very simple approach. So what we need is we need pure protein to get both the training and test data sets. So this is what we do. We purify a lot of proteins, for example, from plants to get them available for the calibration experiments. We characterize the resins that we investigate in our models, for example, in terms of porosity, ligand density, etc. What we also do is we try to establish new descriptors. Descriptors are values that can capture certain properties, certain features of a protein. And again, maybe we can use an analogy. So if you want to create a descriptor, you want to capture, say, the surface charge of a protein, you can use a function to relate that to a simple scale. An analogy would be if we want to characterize, say, a human being or the fitness of a human being, we could use the height and the mass, which may not be that much of a good descriptor because a tall person or a small person could be both very fit or could be rather unfit. In contrast, maybe the body mass index or maybe the distance swum in the last year could be a more indicative descriptor for how fit a person is, and so is our search for descriptors for protein properties. So the isoelectric point in the protein sense is a very simple thing to calculate, the eccentricity as well, but other parameters that describe the charge distribution on a complex surface like this is something that requires some more thinking. What we do in addition to that is we work on faster algorithms to calculate all the parameters and all the models that I just showed you in a meaningful time. So often these calculations can take, say, between an hour, can take hours or days. Now we want to get them into seconds, or we have actually found a way to get these calculations done in seconds to minutes so that we can update and work on the models more quickly. Now this is the major science part, and at the end of this lecture I want just to show you briefly two things. And that is some product examples, and this is, in the interest of time, I'm going to do that really quickly. So one example where it's very useful to use plants as an expression system can be a protein called viscomin A. It's a toxic protein and therefore it cannot be produced in animal cells due to its inherent toxicity, and people have resorted to, say, bacteria to produce it. Because the protein is complex, that is, it has two subunits connected through a disulfide bond and it has internal disulfide bonds, the production process in bacteria is rather complex. So you need to produce the individual compounds of the protein in individual processes. You need to purify them, and then you need to co-refold them. That is, you need to denature them, fully remove the structure, and then refold them in a single process and purify them again. In contrast, if you would do that in plants, you can very much simplify the process because you just have a single extraction, a single clarification, and a single purification process. And by implementing or by switching from E. coli-based process to a plant-based process, we were able to reduce the number of unit operations by 12, cut down the downstream process time from 96 to 7 hours, and increase the activity by threefold, which you can see down here. So the blue curve is a control, so you don't see anything, any toxicity. On the y-axis, you can see the toxicity. So the faster this curve is dropping, the more active, the more toxic is the protein, and this is what we want. If we look at the bacteria-derived protein, the curve is further to the right. That means we need more of the protein, more of the toxin to achieve the same toxic effect as we would with the plant-derived product. Another example is nanoparticles. So here we have used the ultra-diafiltration purification very effectively. Essentially, this is just a single-step purification. We have an overlay of proteins found in the homogenate. So it's full of proteins. Here is Rubisco. Here we have detected our product. We blanch the plant cells, that is, we heat them up, which removes most of the host cell proteins. We filter it, and then we purify through ultra-diafiltration. We enrich our product. And what we can show is that we can use this product, this ferritin protein, to form nanoparticles that can be charged with an anti-cancer drug effectively. Lastly, we also work on total protein purification. Here, what we've done is we used a rather exotic plant, which is called moringa, and we have extracted, or we have built an extraction process based on screw presses, recovering the green juice, heat precipitating the protein in the green juice, and obtaining a pellet. And the good thing about this pellet is that, in contrast to the whole plant, which could be used as a feed for animals, we have removed the saponins, which is a so-called anti-nutrient and preventing the uptake of proteins and other substances from the plant material. So if you look at the pellet, what you can see is that we have substantially reduced the concentration of saponins, making the digestibility or increasing the digestibility of the protein, and at the same time increase the total flavonoid content, which is also, a positive ingredient in this food preparation. Ultimately, this is going to be used as a food or a feed replacement for fish meal. As the last part in my presentation, I would like to briefly talk about integrated biomanufacturing and how this can be implemented. So, from my side, this has two levels to it. On one hand, we have the data integration, and on the other level, we have the mass fluxes that we can integrate. On the mass flux side, I think we have seen that there are a lot of products that we can produce in plants, and this is our primary process. There's some additional products that I would like to highlight. For example, Janos Bindic, one of my postdocs here, is working on a process to produce opiates in plants, and this is going to be, if successful, this is going to be a very large-scale process. So this means that we will produce substantial quantities of biomass in this process that we have to do something with. The good thing is we can use this residual plant biomass and extract other valuable compounds like protein and DNA from it, small molecules, lignocellulose, etc. And specifically, just to give you an example of what has been done and what can be done, I would like to use the a production process for a monoclonal antibody, and the major production process is up here. And for example, in the first chromatography step, as a side product, what you get is the so-called flow-through. This is, in fact, a mixture of proteins and DNA that has been sterile filtered. And what you can do is, or what my colleagues Martin Lobedan and Jörg Schupan have done, is they took this flow-through, subjected it to a heat treatment, and purified a red fluorescence protein from this side product that would have otherwise gone to waste. So produced several grams of red fluorescence protein and produced a lot of plant protein that can be used, for example, for feeding purposes. And the good thing about this is all this previous process came for free, because otherwise this would have just gone to waste. So this is not associated with substantial cost in the process, but is actually making best use of the resources that we have. Similarly, and for the interest of time, I'm just going to briefly mention this, we can also isolate whole protein from different other process intermediates. Overall, plants cannot just be, say, the producing end of residual side streams, but can also be at the receiving end. That is, we can use, for example, waste streams from mammalian or bacterial cell culture and take the spent media and or the homogenized biomass as a fertilizer to foster the cultivation of plants, to make plant cultivation more energy effective. And then ultimately use the plants either to generate novel products like the opiates that I mentioned, antibodies, or to use them for the production of growth factors for mammalian cell culture. But what about the data? Well, on a wet lab side, we are working with high throughput equipment that is generating a lot of data. For example, on the purification of proteins, on the analysis of expression levels, etc., etc. In addition, there is even more equipment that is not on this picture. So we are generating a lot of data. But what to do with this data? At best, we would interconnect all these devices and collect the data so that we can make it accessible for analysis and process optimization, for example, also in the context of improving downstream processing. To achieve this, what we need is we need to interconnect our devices. And we had done this in my previous job, and it's not an easy work to do because you have to think about all the protocols, all the drivers that you need for the different software types. You have to consider all the proprietary data formats. So this is a major effort. The next thing that you need is you need a database that can actually support the data structure that you have. And the data structure is something that you have to develop. So you have to think of what are the different types of data sources in my processor, which reader do I have, which type of device do I have that is creating data. And we need to talk about, or we need to consider the structure of the data files. That is, we have to make sure that in between the different files that we are, or where we are storing our information, the information can be retrieved and can be used for analysis. And to do that, what we have to define is user queries. That is, we need to make sure that we have meaningful questions to the database, like what is the expression levels that we are interested in, what is the process time, what is the yield, etc. And we need to have suitable data analysis tools at hand that can process the large quantities of data that we generate. Ideally, this can be implemented through a multi-layered approach where we have, for example, an electronic lab notebook at the very, say, base level of our experimentation to collect all the data. Then we have a LIMS system that is extracting the most meaningful data from the ELN, and ultimately a scientific data management system providing the high-level analysis that is directing the future research. Ultimately, that approach can support our endeavors in predicting, or going from data from the chromatograms to the prediction of protein separation through, for example, the hybrid models that I presented to you. And with that, I would like to conclude my talk. And I hope I was able to show you some of the ideas and some of the content that we research on. I think there's plenty for us to do. So what we do is we work with plants and plant cells, and we model downstream unit operations, ultimately also to implement a cascading biomass use. And we integrate both the upstream part and the downstream part of the process, because from my perspective, it's not useful to separate processes into these distinct steps, which is creating artificial boundaries, but we have to look at a process as a whole. And then I think I had shown you some examples where we made use of these technologies. And with that, I thank you very much for your attention. And I'm very much looking forward to our discussion. Thank you so much.
So, Johannes, thank you for this very, very informative presentation. I think there's a very good view on that, what you have in mind with respect to your teaching activity and how you want to approach the students. So the nice thing now, I will go for that tool. So we are open for questions, and what else is very important, there is unlimited time. There is no parallel session, so we don't have to keep the time schedule. And whoever wants to be first, we are open now. And also all people participating via the internet connection, we can also go for these questions. So maybe to start, yeah, you showed this very nice drawing, this curve on the technology, on plant technology, and how there was this peak and then it goes down again. So there's a lot of discussion on Industry 4.0, yeah, this digital presentation of the process and all these kinds of things. What is your idea where we are in this context?
That's a very good question, Gerald. I think we're either at the peak or the tip just after the peak. So I think there is a lot of say expectations regarding Industry 4.0, but I think the technology is probably not yet implemented in a way that it's operated in a productive environment. So that is, we would need, for example, technologies to implement augmented reality on the shop floor. And as I've shown on one of the slides in my presentation, there are companies working on that, but it's not, say, the usual thing. So you don't walk into, say, our Technikum and put on digital or 3D or virtual glasses and walk through the equipment and say, okay, now you need to do this, now you need to do this. So this is quite a step, or quite a way to travel until we've implemented that on a regular basis, if that does answer your question. So I think we're at the tip, and we're going to see some disappointment first, because it's not rolled out quickly, and there is some work to do, but ultimately we will go through that valley of disappointment, and once we have the technology available to roll it out in width, I think this is what we're going to see, no doubt about it from my perspective.
Okay, thank you. So my point here is, I think that we have seen already some peaks and downs again. Yeah?
Downs again, okay.
Because
yeah.
this is what I experienced. It's there for a long time already, and it's big. I think we are on a good way. Yeah, at least all what you have shown also with this data processing and all these kinds of things is going to be implemented quite soon. I hope so, yeah?
I think it's going to be implemented, so we will see it, I think.
So, who will be the next? Who is going to be the next? This is
Don't hit the
less
camera.
risk. So, oops.
Should it give it to you?
No.
No?
You're the one, huh?
Oh, thank you. Thanks for this excellent talk, Johannes. And so I have a very general question. So do you expect that plant molecular farming will be just another production system for biopharmaceuticals, or will it, let's say, or will it substitute other systems, or so? What will be the most important selection criteria for using plant molecular farming?
Yeah, very good question. So I take out my crystal ball and make some predictions now. Well, I think it really depends. As I said also in my presentation, I think there's no one system fits all products, right? And for example, if you just look at the transient expression where you need a microbial fermentation at the site in addition to your major process, well, if you can do the same product just in the microbial fermentation, well, then it's difficult to believe that you could be cheaper or more cost efficient with the plant-based process. However, if the product is so complex that you cannot produce it in bacteria and it's toxic so you can't produce it in CHO cells, then this is where plants can go. On the long run, I think, what might be a perspective, and I'm saying might on purpose, is that if we wanted to look at sustainability, and if sustainability is going to get a price, and the price tag to the product, not just to society, so that we as a society pay the, say, the pollution that is being created with some processes, then I think plant molecular farming might substitute a larger fraction of the production capacity in the mid to long term, because in the short term, I don't see that happening, because you need a very different skill set for the what's called upstream production, right? So if major pharmaceutical companies, they have a lot of very well-trained staff that can operate bioreactors. But if we're switching to intact plants, what we need is a completely different infrastructure. So we need greenhouses or vertical farming units, and we need gardeners. We don't need like technicians and engineers that can operate a bioreactor. So this is a transition process, but from my perspective, if we find that quality-wise, plants and CHO cells do the same thing for, say, antibodies, which is a tricky example maybe, but then, yeah, that might replace a substantial fraction.
Okay, thank you.
You're welcome.
Who is the next one?
Rainer?
Thank you. You almost generalized it that proteins which are toxic to bacteria are not toxic to plants. So is that true, or why is that? I mean, it's the metabolism is not totally different, isn't it?
No, then I probably misexplained that. So what I was saying is you can produce these toxic proteins in plants because in plants you have different cellular compartments that allow you to separate the toxic protein, spatially separate the toxic protein from its molecular target. So for example, if you have a ribosome activating protein, and if you target then this ribosome activating protein to the vacuole where there are no ribosomes, so you have sort of neutralized it, so you have isolated it, spatially isolated it, so that it might do less harm. I'm not saying this is perfectly working, because at some point you will have to synthesize the molecule, and if it's escaping the targeting, then you will find some residual toxicity, and this is obviously also what we find in the plant system. There is going to be some residual toxicity, and some proteins might unexpectedly be toxic to plants. So I'm not saying this is working all the time. As I said, you really need to look at the product. And from my perspective, ideally, what we would have in the future is an in silico workflow where we look at the protein, can look at the properties, integrate all the information that we have, and then decide, okay, this and this expression system is probably useful. We test that, and then we choose the one that is working best. So we can exclude some expression systems from testing to streamline the process, and we can focus on those that are relevant and identify that's working best.
By the way, when I was a child, I was trained how to make good spearheads. Not in school, but you know,
Yeah.
you never know.
Very good. I mean, we could do a short quota, like how many people know how to do how to make arrowheads. So you do, but I guess then you're the only person in the room, I would dare say.
So maybe you can forward the
Yeah.
microphone, and I will ask one question from our internet audience. Is there any efficient way to remove the polymers used for flocculation so that you still can perform chromatography?
Yeah, so also a very good question. I would think that really depends on the type of flocculant you're using, right? So if your flocculant is positively charged, and if you're using a positively charged resin as a first capture step, then you won't have any problems. I would claim, at least I haven't seen any, you wouldn't have any problems with that flocculant, and therefore in a subsequent step you will have removed the flocculants and then can continue with regular chromatographic purification. So if you design your downstream process properly, and if you choose the sequence of unit operations wisely, that shouldn't be a problem. But if you want to quantify them, that's rather difficult because they are kind of inert. And at least the feedback that I got from the manufacturers is that they themselves have difficulties quantifying and detecting the flocculants. So if you can avoid them, obviously that's a good thing, but if say, the performance they provide is so much better than what you could get without them, then it's probably useful to use them.
So great talk, Johannes. Here I am.
Yeah.
What about the downstream byproducts you were talking about, the value? How do you calculate the value? I mean, when you're talking about the fertilizer for plants or the red fluorescent protein, can you compare it with something? Because yes, it is valuable, but who are your customers? And it can also be produced much cheaper. And I thought about the urea in the inclusion bodies for solubilization of inclusion bodies. Yes, this is a chemical compound, and this is completely different. But can you comment on this a little bit?
Yeah, that's a very good point. So you're absolutely right. If you wanted to produce just DS red alone, producing it in plants might not be cost efficient. But as you can say, cut the costs for the upstream part, like the plant cultivation, you can cut the costs for the extraction, especially the clarification, and even the first chromatographic purification. So essentially the cost for producing DS red is heat precipitation and IMAC purification. And if you compare that to, say, bacterial process, where you probably need at least IMAC purification as well, so then this is going to be more cost efficient. But the DS Red is just one example. So that was an unintended byproduct for us. Obviously, what you could think of is either producing some other more valuable proteins like enzymes at the site, because what we have seen in our setup, and that was a transgenic setup, was that admittedly the expression level of the protein was moderate, and therefore the expression level was not affected by co-expressing another protein. Obviously, if you're having a very highly expressed target protein, then co-expressing another protein might reduce your yield, and so again you have to look at the specific situation if this is going to be cost efficient and feasible or not.
Thank you very much.
You're welcome.
Okay, so maybe there are also, oh, there's the next one.
Hi, Johannes. Thank you very much for the great presentation. I'm really looking forward to seeing how your future at Boku pans out, particularly on the research side, but also my question's more on the teaching side. When you were discussing how we educate students for the future, you used the term disinvest. How do you see your role as a professor, and more specifically your role as a professor at Boku? Are you here to educate students for future roles in industry, or are you here to inspire passion and a deep knowledge and history of biology? Maybe it's a hard question.
I'll try to answer it. So first, I wouldn't see why preparing students to work in industry, it doesn't mean at the same time that I can't try to be an inspiring professor that is, trying to bring across the spark for curiosity, etc. So this is my personal perspective, if this is answering that part of the question. And yes, obviously, I think that a major part of my job is educating students to find their place in society, in fact. So it could be a job in industry. Personally, my heart beats for science, but that doesn't mean I can't educate people to work in industry. So some of my best friends work in industry, right? And without them, without another support, in addition to the public funding, it would be difficult to do the science. So companies support doing science, and therefore some people have to do this. And admittedly, not everybody's heart is beating for science, but some want to focus on the application, and that's perfectly fine. If we all of us would just do the basic research, the system wouldn't work. So my perspective is that I want to make sure that the students can choose for themselves what they would like best, and that they are best equipped to make that decision, and hopefully that they have a good understanding about what awaits them, like what awaits you when entering industry and what awaits you when entering academic research, to make an informed decision and not just say, oh yeah, I like application, so I'm going to go into industry, because you can do applied science at an industry as well, at a university as well, sorry. So, and regarding the disinvestment, well, I think I have to start with myself, and I have to ask myself, what part of the lecture can I remove? So the feedback from the students that I got was, I have so many slides in my lecture, and that is true. That's a criticism, that's just true. And the next step I have to do is, I have to disinvest, so I have to think, okay, is this important? Or can I put that, say, on the extra slides, on the extra notes, in the FYI part? So if you would be interested in looking at this, and this aspect, I'll give you the additional slides, but we're not going to talk about it right now, because this is, say, and I'm not saying it's not relevant anymore, but from a historic perspective, that and that would be interesting too, or maybe at a more in-depth discussion, but we have to cut at some level and postpone or shift the other parts out of this specific lecture. And that's how you can, structure the curriculum, that you have some lectures that are for everybody. They should cover the basics, and then you have the, say, specific lectures that go in depth and sort out the very specific parts to these lectures. That would be my perspective on the disinvest part. So it's, you have to let go on something you like, and that can be difficult.
good answers. Thank you.
Thank you, Johannes, for a very nice talk. Of course, I liked particularly the overview on molecular farming. And in your last part, you mentioned the cascading biomass use and the side stream product use. Do you think at all, and if so, in which direction, will this influence perhaps the choice of plant species in future?
So I'm not sure if I got the question. So your question is, how will it affect the choice of plant species?
Or will it affect at all?
I think so. I think it will affect, because if a certain plant species does allow for this integration, the business case is better. But again, I think it's a holistic assessment that we need. So if, say, that specific plant is growing faster, but doesn't offer so many side stream potentials, will it still be an overall sustainable process? So it's a very difficult question, and I think it's going to be a case-by-case decision. But ultimately, once we have accumulated sufficient information, I think some plant species will probably crystallize as the ones that are most amenable to that, most compatible with such an integrated bioprocessing. Yeah. That's my perspective.
So then in between there's another question from Fazel Ansori. What are the possibilities for pursuing research among smart manufacturing experts and biomanufacturing ones towards sustainable production systems?
Could you repeat that question?
What are the possibilities for pursuing research among smart manufacturing experts and biomanufacturing ones towards sustainable production systems? So how you can transfer this, what you find out in this field, is this something also useful for sustainable bioprocessing?
I'm still not sure if I have fully understood the question, I have to
Me
admit.
too.
If the question is what we can find using plants to transfer that to other conventional systems, well, I think then yes, definitely this can be done, because at least from my perspective, this is also what we have been discussing here at BOKU with other colleagues, is how can we make use of the side stream potentials that are being generated in, say, bacterial processes or mammalian processes, and that doesn't have to include to use them in or with plants. It can just mean we can recycle them, and it also does mean that we can, say, have sustainable processes without plants, for sure. If that's the question, I'm not sure about that. If the question is more towards how can we transfer our, say, experience that we gain in bioprocessing to other processing technologies, that would be a difficult question to answer, I think. It would probably relate to the question, what is the usability of side streams? So for bioprocess, because it's a biological product, if nothing else comes to mind, you can always burn it, because it's a biological product, it will contain carbon in it, so you can always do something with it. If it's if your waste stream is, say, some metal alloy residuals, then it's probably going to be difficult to make use of it other than sort of remelting it and re-entering it into the process. But I think that might be, we probably, at least I would need to spend some more thoughts on that to give a reasonable answer here, I think.
So there is maybe one, this one here, yeah?
Hi, Johannes. Thanks for the interesting lecture. I was wondering, you outlined some individual unit operations and what you and your team has done to improve them. I would be interested in the perspective across the whole process, kind of what's currently driving the, let's say, the cost-benefit question, or what's really in the next frontier, what really needs to be unlocked in order to further, let's say, drive the cost down, increase the efficiency. If you can say that, let's say, not linked to an individual protein, but more in the grand scheme of things?
Yeah, very good question. I think there's different activities. So my perspective is that in many processes, non-pharma processes, chromatography is prohibitively expensive. So we are looking at one process where we want to purify, say, amino acids for food and feed applications, and then chromatography is just not, cost-wise, not possible. So it would be interesting to come up with a solution here. Membrane technologies might be an option to use as a cost-efficient alternative. But I think what we're also seeing is new modalities in chromatography. Like what I showed you was the packed bed resins, but then there's membranes. There's also now fibers, fiber-based chromatography, and that is also an interesting setting. And maybe a trend is now integrating different unit operations into a single step. For example, with these fiber-based chromatography, what you can do is you can actually do a sort of clarification, lysis, and purification step all in one. That can be interesting. On the other hand, it might be a process step that can be difficult to optimize because there's cross-reactions, interconnectivities in the system. And then I think it's like an up and down. So you tend to go to integrated processes like aqueous two-phase systems is also an integrated process. Or the screw press extraction that I showed you, it's also an integrated process. It's extraction, clarification, and concentration at once, but it's difficult to optimize. And then at one point when starting working with it, you find it interesting. But then if you look at the downsides, you have to look at the specific product to decide, is this going to be worth it? In the end, I think it will help us to create an expanded and augmented toolbox to use for biomanufacturing, for production. And it will be interesting to see if we can keep the overview of that toolbox and if we can, if industry, like the producing industry, like Thermo Fisher, can provide the solutions for these manufacturing processes at a cost-efficient way. Like, are there enough customers to buy these solutions?
Okay, so then there is another question from our online community. Your opinion and forecast on upscaling of BHA, BHB extraction within this decade, so production of polymers. Are you going to invest some resources, focus on this at Boku? Is there any idea from your side to go for that?
That's also a very good question. So I have to admit, my background is in pharmaceutical proteins. So this is a new topic for me. We are starting to work on amino acids now, and this is, say, for me, a very interesting, a very new area. Going for the polymers, the question is it monomer production, or is it really the polymer itself? If it's monomer production, this is going to be tricky, just because of the costs. As I said, for the amino acids, it's the cost pressure is super high, and the purity requirements are immense. If you want to use, this is what I learned so far, so if you wanted to use the monomers for polymerization reactions, you need the 99.9% purity, or at best even better than that, to avoid an abortion of the polymerization reaction. And getting these purities at super low costs is probably challenging. Nevertheless, it would be interesting to look at that, but I think to do that, it would be nice to have an industry partner.
Okay, so as there's no further question from our internet people, there's a nice question that nicely connects to that topic here. So there's a lot of discussion on replacement of food by biotechnology-based products. This is also a huge expectation now that we should solve that big social problem, societal problems. What is your opinion on that? This is also related to our position at the university. For sure, it's nice to have this interest and to get projects in this direction, but what is your own opinion on, for instance, cultured meat?
Very good question. Well, I think yes, as a university we are responsible to work on that. I think it's actually though the second tier of responsibility. The first tier of responsibility would be politics, to make sure, say, that the resources that we have are distributed equally, that people have access to the food that we have. I heard once that the amount of bread that is being put to waste in Rome is the same quantity of bread that is being used in Naples every day. So if we have a situation like this, I think it's difficult to talk about we need to produce more food. I think first we need to distribute it more equally. But then, of course, yes, on the mid to long term, we need to work on alternative production food systems, because I think the way we produce foods now isn't necessarily sustainable. Looking at meat, however, but this is my very personal opinion, and everybody has a different one on that, is personally I would just eat less meat. And I try to do that by cutting meat during breakfast. I don't need that really. When I go to a restaurant, yeah, I would eat meat probably, but then it's okay if it can be a little bit more expensive. I know that that is a very privileged situation, and not everybody can afford that. So who can I be to judge that, right? But then looking from a scientific perspective on artificial meat, my opinion is before we discuss that and put a very large scientific effort into it, we should draw the the boundaries of the balances right. That is, if people tell, okay, this is going to be super sustainable because the CO2, carbon dioxide footprint is much smaller than for, say, conventionally cultured meat. And then if you look at the studies and people draw the boundaries just around the input and output variables, so or say the carbohydrate that is being put in and the meat that is coming out, but not around who did build the bioreactors. What is the cost of producing the growth factors? What is the cost of missing the word. Doesn't matter. But if you don't get the boundaries of the balances right, it's difficult to judge that process. So first, I think we need a very reliable and well-funded assessment of the economic and ecologic input of such processes.
Okay, so thank you. This is very much the same what I think. Okay, so I think it's time now to close the session. There is this surprise package, and I would say it's really that way. It's a sweet one, so we are very happy with you having you here. And I think it's worth to go for another applause. And then there is outside everything prepared. We can discuss there, and we can have fun, and we can have a drink together. So really, thanks for this very, very
Thank
nice
you very much. Thank you. Danke dir. Ja, ja, der ist